Patient is 22, already had myocardial infarction, low-density lipid cholesterol is over 900 milligrams per deciliter. He's got Achilles tendon xanthomas, yellow deposits near the eyes, and corneal arcus.
That's classic familiar hypercholesterolemia.
Wow, diagnosis in under a minute. Why do we even get paid by the hour?
Familial hypercholesterolemia is an autosomal dominant genetic disorder. Normally, low-density lipid receptors in the liver clear the LDL from the blood.
There are three principal causes. Mutations in low-density lipid receptors prevent receptor function. Mutations in ApoB affect ApoB100 mRNA binding, and rarer mutations in PCSK9 cause excess receptor destruction.
So either the liver ignores cholesterol, the cholesterol forgets its name tag, or the receptors get assassinated.
There are two forms. With heterozygous FH, the LDL level is between 350 to 550 milligrams per deciliter. There's premature coronary disease in the 30s of the patient, but with homozygous FH, the levels are between 650 and 1000 milligrams per deciliter, and the patient has cardio events sometimes even before the age of 10.
And our lucky guy, homozygous, the cholesterol jackpot. Anyone have any guesses on how to treat this?
First slide is statins. They boost low-density lipid receptor expression.
But in homozygous FH, statins don't work well. No receptors to regulate, remember?
Other options include ezetimibe, low-density lipid apheresis, and newer PCSK9 inhibitors like laridasilbep.
Basically glue and a prayer. Great medicine.
What about Mipomersen?
1977, Paul Sebastian invents the antisense concept. That's using short DNA or RNA sequences to target messenger RNA to affect protein synthesis, for those of us who didn't pay attention to molecular biology chase. In 1998, the first antisense drug familial is FDA approved to treat CMV retinitis, made by Ionis Pharmaceuticals.
Then, Isis Pharmaceuticals, now Ionis, created second-generation antisense oligonucleotides with phosphorothioate backbones and 2-methoxyethyl modifications. This made the antisense drugs more stable, more potent, and less toxic.
By 2008, the FDA granted orphan drug designation to Mipomersen for homozygous FH due to its rarity and lack of treatments.
In 2008, Genzyme licensed it from Ionis with a $175 million upfront deal. In 2011, an EMA application was filed, and then in 2012, an FDA new drug application was submitted. Finally, in 2013, the FDA approved it under the name Kynamro.
Basically, Kynamro works by targeting the mRNA that produces the ApoB protein.
That's the protein that allows LDL molecules to form in the bloodstream. Kynamro can decrease the level of LDLs in the bloodstream by degrading the ApoB mRNA, preventing the protein from forming.
What are we waiting for? It sounds perfect.
There's a tiny detail of Mipomersen's target being the liver. That means hepatotoxic side effects like liver steatosis, fat buildup in the liver, can occur. The drug was rejected for the European market and is currently stalled in stage 3 clinical trials in the US.
It seemed so promising at first. What happened?
Well, the preclinical trials started off well on animal models, like mice, with them showing a reduced total cholesterol and LDL with overall improved cardiovascular health. Even phase 1 clinical trials, the double-blind placebo study confirmed the expected cholesterol decreases, while the effects on the liver remained asymptomatic. But they did detect increased liver transaminases. The alarm bells didn't go off just yet. It's during the second and third phases of the trial that the adverse effects became more apparent, with a substantial amount of participants experiencing adverse effects due to the hepatotoxicity.
Doesn't seem super relevant.
Home, this is serious. We can't sacrifice the patient's long-term health. There's a reason that the European Medical Agency outright rejected it. There are even reports of injection site reactions in both clinical trials. That's a high chance our patient will get liver problems.
I agree. It'll be safer to at least start on the statins until we can get approval to use Mipomersen.
I think we should use the Mipomersen. Statins have poor effectiveness for familiar hypercholesterolemia, and the patient could suffer from more coronary diseases while we wait.
That settles it. I'd pull rank at Kamarov's backing me up.
There's gotta be a better way. It's completely unethical to bypass regulations to use it. Only doctors in the restricted use program for Mipomersen can use it, which last I checked was none of us.
I have an idea. Remember how PCSK9 enzymes break down LDL receptors?
Yeah, it causes more LDLs to accumulate in the bloodstream because the PCSK9 enzymes break down the liver receptors.
So why don't we supplement the Mipomersen with PCSK9 inhibitors to compete with or destroy the enzymes? The LDL receptors on the liver would come back online and reduce LDLs in the blood. That would allow us to use less Mipomersen and reduce adverse effects.
It might work, but it's still under regulation. If something bad happens, it's on you, home.
I'll deal with that. Start the patient on Mipomersen and make sure to supplement with PCSK9 inhibitors. Also, make sure to monitor transaminase levels. If the patient shows too much, that means the liver is leaking intracellular content into the bloodstream.